Thank you very much, Dr. Smith. So as Kim said, my name is Jim Smith. I am the Director of the Office of New Drug Policy and the Office of New Drugs at FDA. And during this session, I'll be speaking for the next 45 minutes about the basics of clinical trial design. The learning objectives are to describe the characteristics of adequate and well-controlled studies, to describe the purpose of control groups and various types of controls, and to describe methods to reduce bias in clinical investigations. So to begin, and this might seem really basic, but we should keep in mind that the purpose of doing clinical research is to ask important questions and to answer them with appropriate studies. So the quote on this slide is the first sentence of the regulation that describes adequate and well-controlled studies, which are the characteristics of studies that should be submitted to FDA to demonstrate effectiveness for a drug. And it leads right off with a purpose. The purpose of conducting clinical investigations of a drug is to distinguish the effect of a drug from other influences, such as spontaneous change in the course of the disease, placebo effect, or biased observation. And although this regulation was specifically written to address, investigations seeking to demonstrate effectiveness of a drug, like I mentioned, the same principle applies to any study, frankly. One is generally interested in discerning the effect of some independent variable itself, like here the drug, on one or more dependent variables. That is the outcomes of interest. And if something other than the intervention being studied could have plausibly caused the outcome, then the study will have failed to answer the scientific question. So here I'm going to summarize at a high level the characteristics of an adequate and well-controlled study, which, as I just said, are good scientific principles to consider regardless of the type of clinical research that you're involved in. And I'm going to go into several of these points in the coming slides, but let me run through the list as an overview. First, studies should have clear objectives. So these should reflect the research questions, they should be clear and explicitly stated in a protocol, along with a summary of the methods that will be used to analyze the data to answer the questions. The study should use a design that permits a valid comparison with a control group, and that provides a quantitative assessment of the effect of the intervention. So I'll talk about examples of control groups shortly, as well as some common designs that are used to compare two or more groups in various ways. The selection of trial participants should be done in a manner that provides adequate assurance that they either have the disease or condition that's being studied, or in the case of a drug or intervention intended to prevent or reduce the risk for some outcome, the participants should at least be susceptible to having that outcome. Otherwise, there'd be really no reason for them to be subjected to the drug being studied. The method of assigning patients to the treatment and control group should minimize bias. This is where randomization usually comes into play for studies with a concurrent control, which I'll discuss in a bit. And beyond the method of treatment assignment, adequate measures should also be taken to minimize biases on subjects, observations, and analysts. Blinding is an example of such a measure. Next, there should be well-defined and reliable assessments of how the individuals in a trial may respond to an intervention. That is, what outcome or outcomes are being measured? Are we certain that we're measuring what we intend to be measuring? And last, there should be analyses of the results of the study that are adequate to assess the effects of the drug. These should generally be pre-specified in the protocol or statistical analysis plan before the study begins. So this may seem obvious, but it really can't be overstated. The scientific questions to be answered and the trial objectives need to align. So without having a really clear, specific question or set of questions, the study design choices are likely to be suboptimal, and then its results might be insufficient to answer the questions that you're trying to answer. So the questions that are driving the research should really inform choices about trial design data collection, and analysis. So for example, if a new drug is being compared to a drug already on the market, is your question whether the new drug is clearly more effective than that existing therapy? Or would you also be interested in knowing whether it's at least not materially worse than the existing therapy, even if it's not clearly better? So each of those questions would not only require distinct analysis plans, but it would also impact sample size and possibly the importance of blinding and endpoint selection and other considerations. Or as another example, is the objective to understand the effective treatment when it's added to a specific background treatment, or would it be sufficient to understand its effect when it's added on to standard of care, which may or may not include a certain background treatment? The choice of a control group is always a critical decision. And control groups allow discrimination of patient outcomes caused by the test treatment from outcomes caused by other factors. So if the course of a disease were always the same in a given patient population, or if it was completely predictable from patient characteristics, then the observed outcomes that occur in a single treated group of patients, that is like a single arm trial, could simply be compared with the outcomes that you know would have occurred in the absence of that treatment. And that is possible in certain circumstances, but typically a concurrent control group is needed because it's not possible to predict outcomes with adequate accuracy or certainty. And the choice of which control to use is important because it will affect the inferences that can be drawn from the trial, potentially the ethical acceptability of the trial, the degree to which bias can be minimized, the type of participants that can be recruited and how quickly, the types of endpoints that could be studied, the credibility of the results, and so on. So here are the 5 types of controls that are generally recognized in adequate and well-controlled investigations and are mentioned in the regulation that I cited. The first 4 are usually concurrent controls. That is, the control group and the test group are chosen from the same population and treated as part of the same trial. So the first, which I'm sure you've heard of, is the placebo control. So in a placebo-controlled trial, participants are randomly assigned to the test treatment or to an identical appearing treatment that does not contain the test drug. And it's very important to recognize, and sometimes this seems to be misunderstood, that the use of a placebo does not imply that the control group is untreated. In many placebo-controlled trials, the new drug and the placebo are each added to a common background therapy or other standard of care regimen. These trials are almost always double-blind, meaning that the patients and the investigators don't know what the treatment assignment is. And combined with the use of randomization and blinding, which I'll discuss a little bit more in a bit, this design actually controls for many, if not all, potential influences on the outcome other than those arising from the drug itself. So, for example, many diseases wax or wane in severity. So if a participant improves significantly after randomization, was that destined to happen regardless of the intervention, or, just because of the natural course of the disease, or was that a drug effect? Having a placebo arm can give you information about that. Or you might have heard about regression to the mean, where patients are eligible for a trial because they have, let's say, high blood pressure. So a high blood pressure reading gets them into the trial, but that might have been kind of a random high for them just from day-to-day variability. And in those cases, over time, even if without treatment, their blood pressure would usually fall back to that patient's average. And so having a placebo group can help assess for that and can show, what would have happened to a group in the absence of treatment. The second example is the no treatment control, and this is exactly what it sounds like. Participants would be randomly assigned to test treatment or nothing at all. That is not even a placebo. So really the main use of this design is when it's difficult or impossible to blind the treatment assignment. In the third example, participants are generally randomly assigned to different doses of the same drug or different regimens of the same drug. If a higher dose of the drug is shown to be more effective than a lower dose, that can provide really strong evidence that that higher dose is effective. The limitation, of course, is that the result would tell you something about that high dose, but if you haven't also included a placebo arm, you won't know whether or not the low dose is effective. Or similarly, if you don't see a difference between the high dose and low dose, then you're left wondering, are they both effective or are they both ineffective? For these reasons, it can be beneficial to include a placebo arm, which then provides information about a dose of 0, if you will. Next, in an active control trial, participants are generally randomly assigned to either the test treatment or a known effective treatment, which is referred to as the active control or the positive control. These trials can be double-blind by having participants in each arm receive not only their assigned treatment, but also a placebo version of the other arm's treatment, and that's referred to as a double dummy technique. So blinding in these trials is not always possible, though, because of some combination of either the number of interventions or different regimens or different routes of administration and so on. Last, in an externally controlled trial, a group of participants who received a test treatment are compared with a group that is external to the study. So maybe it was a group of patients observed at some time in the past, which is then referred to as a historical control, or it's a group observed around the same time, but in a different setting entirely, like a disease registry or a natural history study, or information collected from medical practice, and so on. Now, on occasion, an effect observed in a single arm trial is known without question to be something that would not have occurred spontaneously. And so although this is sometimes called a baseline controlled study because a value on treatment is compared with the individual participant's baseline value, that's actually also a type of external control, because what you're really doing there is you're relying on the very strong knowledge, which is derived external to the trial itself, that the change that's been observed would not have occurred in the absence of the intervention. And there is a lot of interest right now in externally controlled trials, but it's really beyond the scope of my presentation today to cover them in sufficient detail. Suffice it to say that the lack of an internal concurrent control arm and randomization and blinding require exceptionally careful consideration of various ways that bias could be introduced into the trial and whether it's even possible to sufficiently mitigate these limitations to result in an interpretable trial. Okay, so moving on to the study population, that is, which patients are going to be included in the trial. So typically, the eligibility criteria or the inclusion and exclusion criteria should identify patients with or at risk for the disease or condition being studied. And this population might be more narrowly defined or more broadly defined depending on the objectives, which could vary during drug development. So early stage drug development, perhaps the population is being more narrowly defined because you want a more homogeneous population to detect a signal of a drug effect, or perhaps there's too much uncertainty about the safety of a drug in the early stages of drug development that you need more exclusion criteria. But as that drug progresses to later stages of drug development, it becomes increasingly important to define the study population more broadly to more closely represent the diverse populations for which the drug is intended. And in general, you should avoid unnecessary exclusion criteria. So both to facilitate enrollment as well as generalizability of results, like each exclusion criterion should be very well considered and believed essential. And you should think about even commonly used exclusion criteria if they're actually necessary. Why exclude patients with renal impairment if the pharmacokinetics of the drug have been determined for those patients? Maybe they could be included, but with a drug with a dose adjustment, for example, or an age-related exclusion should be very well justified. So overall, the population that's expected to benefit from answering the research questions of interest ought to be represented in the clinical investigation, and enrollment goals should be proactively set to achieve that. So related to the study population, I'd like to briefly touch on the concept of enrichment. So this is the selection of a study population in which detection of drug effect, if one is present, is more likely than it would be in an unselected population. So this is a design feature that narrows the study population in certain ways in hopes of improving the ability to detect the effect of a drug. And so I'll go through three broad categories of enrichment strategies. So first are strategies to decrease variability, because reducing variability often makes it easier to detect a signal among the reduced noise, if you will. So one could aim to reduce between patient variability or within patient variability. And an example of the latter is what I've included here. So a placebo lead-in period could be used in which all patients are provided a placebo for some amount of time before randomization. And those who show a large response to placebo, or those who demonstrate that they're very poorly adherent to taking the assigned placebo, could be excluded before they're ever randomized, as those participants might be even, might be more likely to introduce variability during the trial, making it more difficult to identify a drug effect. Another enrichment strategy is prognostic enrichment. So the question here is, who's most likely to progress or have an outcome of interest? So let's say you're conducting a trial in which the outcome of interest is hospitalization for heart failure, and the trial is designed to run until a certain number of such hospitalizations have occurred. Well, it could be quite beneficial to enroll patients who are at greater risk for hospitalization, like maybe those who have had a hospitalization for heart failure within 1 year of enrollment, or some other characteristic that informs their prognosis, which is why it's prognostic enrichment. The last strategy I'll mention is predictive enrichment. So here, the question is, who's more likely to respond to the drug than others without the factor that's being used for selection? So prognostic enrichment considers who's most likely to have an outcome regardless of treatment. And predictive enrichment considers who is predicted to respond to the drug. And so a randomized withdrawal study is an example of this. So here, all of the patients are placed on the drug for some period of time, and then a certain subset of those who believe they've responded to the drug by some predetermined criteria are then selected to be randomized to continue on the drug or to switch to a placebo. And that's an enrichment strategy because all of the patients who don't think that they're responders to the drug will have been weeded out, if you will. And they won't then be subjected to the test versus control randomized portion of the trial. Another example of predictive enrichment, maybe it's even more straightforward, is let's say you're enrolling patients who have a tumor tissue that's positive for a certain biomarker, such as a certain gene variant, and the drug's mechanism of action specifically targets that gene variant. That would be another example of predictive enrichment. Enrichment strategies can limit the generalizability of the findings, and I think Dr. Levinson might touch on this in his talk that's coming up. Okay, next we have method of assignment to treatment arms or study arms. This should be done in a manner that minimizes bias and ensures comparability of the groups with respect to pertinent variables. So typically, in a concurrently controlled study, assignment is by randomization. So why is randomization important? Let's think about a couple scenarios here. So in a randomized study, a patient who meets the eligibility criteria gets assigned to a group by a completely random method. So I'm illustrating that here by a flip of a coin, if you will. So heads, they get drug A, tails, they get drug B. So coins aren't actually used, but I think you get the point. So now think about an observational study where you go to the medical records of patients who are getting drug A and patients who are getting drug B, and you record things that you find in the record about their medical course, and then you compare the outcomes of interest of those who got drug A versus those who got drug B. So, well, why could that be a problem? Well, just highlighting one concern for illustration of this point, consider how these patients each came to be on drug A versus drug B. So they may have come to their healthcare provider who took a whole host of things into consideration when making a treatment decision. Maybe the patient's age, their comorbidities, concomitant medications, family history, labs, the patient's own preferences, their insurance status, and so on. And if a difference observed between outcomes for drug A compared with drug B, if a difference is observed between those two, it's plausible that some other factor or factors other than the drug itself is associated with the selection of which drug to prescribe, but also with the outcome itself. So this could lead to an incorrect conclusion that there's a difference between drug A and drug B, when in fact, the difference is the result of the underlying patient populations. And this is referred to confounding, and I think that Dr. Levinson might be discussing this as well. So assuring that the subject populations are similar in the test and control groups is best attained by randomly dividing a single sample population into the test and control groups. So randomization is going to avoid systematic differences between groups with respect to both known and unknown baseline variables that could affect the outcome. And the balance of unknown baseline variables is an important one to keep in mind. It's not uncommon to see a great deal of hand-wringing over various baseline characteristics and their apparent balance or lack thereof in a randomized trial. And sometimes that is justified, especially when there's this known strong prognostic or predictive characteristic. But often it's not. In part, it should be kept in mind that there are a very, very large number of unmeasured baseline characteristics, and we have zero idea about their balance. Or lack thereof, because we have no idea what they, or their values are. Of course, this doesn't mean don't trust a randomized trial. In contrast, appreciate the ability of randomization to avoid these systematic differences between groups. And perhaps even more important, recognize that randomization provides a sound basis for statistical inference. And the reasons for that are beyond the scope of this presentation. But it really is a very important outcome or a reason for randomizing participants in a trial. So it's important that the control group be as similar as possible to the test group, except for the intervention that's being studied. To best assess the effect of the drug itself, the similarity between groups should be both at baseline as well as after baseline, through the study. And as I just discussed, randomization facilitates the similarity between groups at baseline. But many differences can arise after baseline, differences in adherence to assigned treatment, the use of rescue therapy perhaps, completeness of data and follow-up, and differences in how the outcome assessments are conducted, reported, and so on. And in the next few slides, I'll talk about methods to reduce bias entering the study during the trial itself. One of the key methods to reduce bias, at least in a randomized study, is to conceal the treatment assignments, which is referred to as blinding. So blinding is intended to minimize the impact of potential biases that could be conscious or unconscious. And as I just said, the aim here is to ensure that the study groups are treated and observed similarly during the trial, except for the intervention of interest. So what impacts could the knowledge of treatment assignment have? Well, I'll give several examples on this slide, but it's certainly not all inclusive. And I'm also not saying that these will arise in every open label trial, but these are the types of things that should be considered and mitigated if possible. So healthcare providers could treat the trial participant differently if they know what study arm they've been assigned to. And those differences in how they're treated could directly affect the outcome of interest. So this would be another example of confounding. Those on active drug might report more favorable outcomes, such as responses on a questionnaire because they expect they should improve on the new drug being tested. Observers may be less likely to identify and report treatment responses in the control group, like the no treatment group. Those on active drug may be more or less likely to report adverse events. And this is a good reminder that oftentimes you don't know which direction these biases may go. Right? It's unpredictable. Knowledge may affect rigor of attempts to obtain follow-up data. And knowledge could impact treatment decisions related to analysis. Now, this isn't the investigator or the patients, but this could be a way that knowledge of treatment assignment could affect those other personnel, like those who are analyzing trial data. So that could impact their decisions, such as looking for reasons to justify excluding a poorly performing site. If getting rid of the results might make the rest of the trial results appear much more favorable for the test drug. And as I mentioned, there's no simple test for any of these. You can't really look after the fact and say, oh, well, this bias must have crept into the trial. And sometimes it'll favor the test drug, sometimes not. And you can't really assume that that's going to all balance out. So taking measures to reduce bias, especially in circumstances when blinding isn't possible, is really important. Now, even with blinding, it's important to realize that patients or investigators might be able to detect or guess their treatment assignment. Maybe there's a distinct adverse effect, that's only observed with the test drug, for example, or something similar to that. And in such cases, especially if the study is open label, one should consider pre-specified decision rules, blinded outcome assessments, and other measures. So as an example of a blinded outcome assessment, if the endpoint of an open label trial requires the histological assessment of tissue, or maybe the rating of a skin lesion from a photograph, the individual conducting the assessment should not be made aware of the treatment assignment. And this doesn't prevent all bias from entering the study, of course, but it would be an attempt to mitigate a biased assessment. Again, whether conscious or unconscious, by the study personnel who are aware of what treatment the participant is receiving. Knowledge of interim results, whether it's at the level of the individual or the treatment group, also has the potential to introduce bias or otherwise influence the conduct of the study and interpretation of its results. So special attention should be given to maintaining the confidentiality of interim results. And finalizing statistical analysis plans before treatment assignments are revealed is another mechanism to ensure that knowledge of the data itself does not influence the choice of analyses, or the choice of the inclusion or exclusion of certain data, the weight that's placed on certain endpoints, and so on. So shifting gears a bit, next I'd like to talk about assessing response in a clinical investigation, which is often described as the endpoints of the study. So the choice of endpoints should be aligned with the study objectives. And if the study is being conducted to answer the question of whether a drug is effective for a certain disease or condition, those endpoints should be meaningful for the intended population, and may also take into account the views of patients. The primary endpoint should provide relevant evidence related to the primary objective of the study. So, and typically the sample size of a trial is calculated based on information related to this endpoint. And then secondary endpoints are usually either supportive measurements related to the primary objective. That is, they provide additional information that might give one greater confidence in the results observed on the primary endpoint, or they may be measurements related to secondary objectives of the study. Usually the primary endpoint and at least some secondary endpoints are included in an analysis plan that protects against making false conclusions that an intervention has an effect when in truth it does not. Exploratory endpoints are those that may further explore or support study findings, or they may explore and generate new hypotheses for future research. So exploratory endpoints would generally not be included in the analysis plan in a manner that protects against making false conclusions. And because many potential endpoints and analyses could be explored, there's the very real chance that a difference between groups could be observed on one or more of these endpoints solely by chance alone. But what is an endpoint exactly? So this is a definition that comes from the current version of the FDA NIH BEST resource. BEST stands for Biomarkers, Endpoints, and Other Tools. So an endpoint is a precisely defined variable intended to reflect an outcome of interest that is statistically analyzed to address a particular research question. So when thinking about an endpoint, obviously you need to identify the assessment itself. For example, is it a lab value, a patient-reported outcome like a questionnaire, a physiologic test like maybe a pulmonary function test, or a clinical event like a stroke. Next, the endpoint might incorporate other information that occurs during the trial. So for example, if the endpoint is defined in terms of a success or failure, perhaps participants who discontinue their assigned study treatment prematurely are automatically considered a treatment failure. It should consider the timing of the assessment. So for example, are you interested in a hemoglobin value after 6 months of treatment? Or maybe the assessment is one that could happen at any time, such as a participant having an event like a stroke, and really the time of interest is the duration from randomization until when that first stroke occurs. And finally, if relevant, consider how assessments within each participant are formulated or combined. So for example, sometimes a composite endpoint is used, which means that multiple distinct events can be combined, if you will, into a single endpoint. Or maybe several assessments are made during the window of time, like a daily diary over a 2-week period. And you might decide in advance that you will take the average of all 14 scores, or maybe it's the maximum or the minimum, to represent the endpoint assessment in a single value. So there are different types of endpoints, and I've already alluded to several, but a couple major categories include clinical outcomes and surrogate endpoints. A clinical outcome is an outcome that describes or reflects how an individual feels, functions, or survives. That is, it directly represents the clinical state of an individual. A surrogate endpoint, on the other hand, is an endpoint that is used as a substitute for how a patient feels, functions, or survives. So an effect on the surrogate endpoint is expected to predict an effect on the ultimate clinical outcome of interest. And the level of evidence that an effect on the surrogate predicts such a clinical benefit can vary, of course. So if the evidence is strong, at least in drug development, that's called a validated surrogate endpoint. Examples of this could include reductions in hemoglobin A1C, which have been clearly shown to predict benefit with respect to microvascular complications and diabetes, or reductions in blood pressure, which reduce the risk of certain adverse cardiovascular events, such as stroke, among patients with hypertension. I'd also like to mention the idea of intercurrent events. So intercurrent events are events that occur after treatment initiation that affect either the interpretation or the existence of the measurements associated with the clinical question of interest. So some examples of these types of events could be treatment discontinuation, and that can occur for a variety of reasons, like adverse events, perceived lack of efficacy, other things. Beginning a new therapy or switching to other treatments, and which could affect the outcomes of interest, or even something like death. And it's important to note that intercurrent events do not lead to a problem of missing data. So missing data is data that would be meaningful for the analysis, but were not collected, which is different than data that do not exist or data that are not considered meaningful because of an intercurrent event. So for example, although missing data strategies is beyond the scope of what I'm talking about in this session, imputation techniques are often used to impute values for those that should have been collected but weren't. So consider a participant who dies during the trial before they reach the time point at which the endpoint would be measured. Well, they won't have an endpoint value, but it would usually not make any sense at all to impute a value for someone who died, essentially pretending that they were alive and had an endpoint measured. So in this case, death is an intercurrent event. And if it's predicted to happen with some frequency in a certain study population in a trial, how to handle such an intercurrent event should be considered and incorporated into the design and analysis plan before the trial even begins. So you should also consider possible intercurrent events when defining the clinical question of interest. So here's an example. In a trial of a drug for type 2 diabetes, suppose the primary endpoint is the change in hemoglobin A1C from baseline to week 24. And suppose the trial allows for rescue medication if certain thresholds are met with respect to blood glucose values being too far out of control, for example. So here are two examples of possible treatment effects that may be of interest. What's the effect of the new drug if rescue medication is not available? Or what is the effect of the new drug regardless of whether rescue medication is used? Now, I don't have time to discuss how the effect of interest would result in different design considerations, but to suffice it to say that these are distinct questions, right? And that should be considered on the front end, not after the fact. Okay, so the elements that I've discussed can be put together in various ways, and I'll just touch on a few here. The most common study design for a randomized controlled trial is the parallel group design. So in this design, participants are randomly assigned to different groups that are then followed in parallel, if you will, until the endpoint. Another study design, which still employs randomization, is the crossover design. So here, participants are randomly assigned to an initial study treatment, so test versus control in my illustration here. And after some period of time, endpoints are assessed. And then there's usually a washout period where no treatment is given, allowing the effects of the first period to, possibly return to their baseline values. And then within each of the initially randomly assigned groups, each participant then receives the other study treatment for a second period, and the endpoints are assessed again. And this design can be more efficient than the parallel group study and therefore require fewer patients because it has the advantage of each patient serving as their own control, which removes the between patient variability from the comparison between treatments. But there are several things to consider that might make this a suboptimal design. So first, there's the possibility of a carryover effect, which is the effect of the treatment from the previous time period carrying over and influencing the response during the subsequent time period. And that washout period in the middle is an attempt to mitigate this, but it's still possible that the previously administered treatment may have altered the individual in some way, so that they'll react differently to the treatment from that time onward, or maybe the washout period is just, an insufficient amount of time or whatnot. Or maybe the outcome is some sort of a functional test that requires some skill, so that participants are typically perform better during period 2 on the task because they were trained in a sense during period 1. So those are things to watch out for. Second, the design is generally used when the observation periods are relatively short, so that the endpoint needs to be one that responds and can be measured fairly quickly. Also, since ideally each individual would return to their baseline condition before each period, it's generally used in chronic diseases where the outcome is not dependent on the time of the assessment. So last, I'll describe a 2x2 factorial design. So I've drawn the study arms here as a table, but this would typically be a parallel group study. But each participant gets randomly assigned, so in this case, to either placebo, drug A alone, drug B alone, or the combination of drug A and drug B. And of course, this could be blinded using the double dummy technique that I mentioned earlier. And this allows you to essentially study 2 interventions using the infrastructure of one trial. So first, you can assess the effect of drug A by comparing the 2 groups in the left column, that is placebo and drug B, with the 2 groups that include drug A, that is drug A alone and drug A plus drug B. Similarly, you can assess the effect of drug B by comparing the top row, so placebo, placebo and drug A, so it does not include drug B, to the bottom row, which does include drug B. But the design also allows other analyses. So for example, you can look at whether drug A and drug B each contribute to effectiveness when they are used in combination. You can also check whether the effect of drug A is modified in the presence of drug B, and vice versa. Factorial trials can certainly include more than 2 interventions, but it can quickly become infeasible since the number of cells in a full factorial trial will simply become too large. Also related to the comparison between groups, and I've mentioned this already, is the question of whether you're testing a superiority or a non-inferiority hypothesis. So in a trial of drug A versus a known active control drug B, the superiority hypothesis is testing whether drug A is better than drug B. And by better, this would usually mean more effective, although I suppose you could test a hypothesis related to superior safety as well. In contrast, the non-inferiority hypothesis would be testing whether drug A is not meaningfully worse than drug B. And the non-inferiority margin, or NI margin, quantifies what is meant by meaningfully worse. Now, if no difference is detected between drug A and drug B in a trial, then there's a question. Does that mean that both drugs are effective or that both drugs are ineffective? And that's obviously a very important difference. And so because there's no confirmation, within that trial itself that the active control drug B is effective, that's an assumption. And so it must be considered very carefully. So assay sensitivity is the ability of the trial to have detected a difference between treatments of a specified size. So said another way, assay sensitivity means that, had the study included a placebo, the active drug would have been shown to have the effect that was assumed. And this is not directly assessed within a non-inferiority trial. So you need to consider such things as, historically, have consistent findings been observed with the active control, especially when compared against placebo in those past trials? If there has been a consistent effect, then you have greater confidence in the active control in the new trial. Second, how similar is the new trial to those historical trials where the active control drug was tested against placebo? Is the patient population similar? Were there important differences in concomitant treatments that have become available? Are the differences in how the study endpoints are defined or analyzed? Is the active control being dosed similarly in the new trial as it was in the historical trial? So these are things to consider because you're making an assumption that the active control's effect is constant between the new trial and the historical trials. And that's referred to as the constancy assumption. Now, third, the quality of the new trial needs to be excellent. So poor conduct can reduce the ability to detect differences between groups because sloppy conduct generally introduces variability. So this can be problematic in a non-inferiority trial because the trial could be considered a success if the new drug and active control appear to behave similarly. And you won't know for sure whether that means that both drugs are effective or both drugs are ineffective if the trial was not conducted well. Finally, just a couple high-level comments about trial design and to some extent conduct. So efforts should really focus on activities that are essential to the study. So for example, it's far more important to expend energy and resources to ensure that the primary endpoint is assessed exactly as intended, that follow-up is as close as possible to 100%, rather than pouring resources into ensuring that every scrap of source data is available for the 50th exploratory endpoint. Obviously, I'm giving an extreme example there, but I'm just trying to emphasize that you should consider eliminating non-essential activities in data collection to increase quality by simplifying conduct, improving the efficiency of the study, and to really target resources to the critical areas. So deploy your resources to identify and prevent or control errors that matter. And last, Clinical study design is best informed by input from a broad range of interested parties. So patients, patient organizations, study coordinators, site staff, clinical investigators, like these, people who will be directly involved in the successful completion of the study are very well positioned to inform its design. Um, Patients may take one look at a protocol and say that it's far too burdensome in a variety of ways, and that could potentially foreshadow a lot of premature treatment discontinuation if it's not modified. Clinical investigators and site staff could give a lot of insight into the feasibility of enrolling patients who meet the inclusion and exclusion criteria, and maybe those need to be modified or broadened. Or whether the scheduled study visits and procedures are overly burdensome, and might also lead to early dropouts. So, and patients and patient organizations can inform the general relevance of study endpoints, study settings, and some among other things. So seek this information out and really consider using it to optimize the design of the study. So with that, I'd like to move on to my challenge questions. The first is, which of the following is not an example of enrichment? Enroll patients with a lab value that indicates they are at greater risk for the outcome of interest during the trial. Limit enrollment to patients with a certain genetic variant that is specifically targeted by the investigational drug. Ask trial participants who demonstrate poor adherence after randomization to withdraw from the trial. And require patients to have 3 consistent values during the screening period for the assessment of primary interest in the trial. Give you a few seconds to think about the answer. And C is not an example of enrichment. So A is an example of prognostic enrichment, B, predictive enrichment, D is a way to reduce variability, but C says to ask participants who demonstrate poor adherence after randomization to withdraw from the trial, and you do not want to do that because after randomization, pulling participants out with poor adherence could introduce bias. And so if you're going to use this technique, it should be done prior to randomization. The second challenge question, the choice of a control group affects which of the following? A, the inferences that can be drawn from the trial. B, the ethical acceptability of the trial. C, the degree to which bias in conducting and analyzing the trial can be minimized. D, the scientific credibility and impact of the results. And E, all of the above. And hopefully that's clear that that should be E, all of the above. So to summarize this session, you should design clinical investigations to distinguish the effects of an intervention from other influences. Always start with the clinical questions of interest, and the design of the clinical study should flow from those. Key design aspects include the study population, an appropriate control, well-defined endpoints, measures to minimize bias, including the assignment of treatment, and the analytical plan. And with that, I'd like to thank you very much for your attention, and I will turn it over to Dr. Levinson.